Hi, my name is Paul Song. I'm the CEO of NKGen Biotech. We are a clinical stage company providing innovative natural killer cell products for advanced cancer and neurodegenerative diseases. Today, I wanted to share with you a little bit of the work that we are doing with our natural killer cells for neurodegenerative diseases, as well as the scientific rationale behind its use. I just want to give you a little bit of background first on natural killer cells. They're probably the most underappreciated cells in the human body. They represent about 5 to 20% of all the circulating lymphocytes, which are the white blood cells in our body. For all the attention that has been placed on T cells, as well as regulatory T cells, I think it is becoming clear that natural killer cells play an equal role in overall regulation. But perhaps the biggest thing that natural killer cells can do is distinguish self from non-self. So those dangerous cells that are circulating within a human body, whether they be cancer cells, whether they be virally infected cells, or in many respects, some dead tissue, natural killer cells have the ability through a whole array of receptors to distinguish self from non-self and help to eliminate these dangerous cells. In fact, if you were to go back and look at the literature on the role of natural killer cells and look specifically at natural killer cell dysfunction, so either weak or decreased numbers of natural killer cells, you will find numerous publications of correlating weak natural killer cells with various diseases ranging from cancer to autoimmune diseases to some neurodegenerative diseases as well. So when we set out to establish our natural killer cell program, we were looking well beyond oncology to really look at the role that natural killer cells play across numerous disease paths. And in doing so, we wanted to create highly enhanced, highly active natural killer cells without any genetic modification that could be used to address not only oncology, but all sorts of other diseases as well. The challenge, though, of natural killer cells are they're very, very difficult to get to grow. Most companies that are in this space have gone to a universal donor, whether it be a pluripotent stem cell or fetal-derived cord blood as their source, because if they were to take 10 healthy volunteers, isolate the natural killer cells and try to get them to grow, most companies can probably get 20 to 30% of all of their donors' cells to successfully grow. So because of the lack of ability to get routine growth, that's why they've adopted a universal source of cells. While that may make sense for an allogenic program for oncology, we felt that that would be problematic to treat non-cancerous conditions. So the first thing that our technology allows us to do is to get anyone's NK cells to grow, regardless of how heavily pretreated the donor is, how old the donor is, or how healthy the donor is, that when we take 100 patients, we're able to get 100% natural killer cell growth. So that's the first thing. The second thing that I think a lot of companies don't talk about is taking a weak natural killer cell and making it inherently stronger and more active in its ability to recognize self from non-self. So not only can we get anyone's NK cells to grow, but we will show you how we can make them inherently more powerful and more in tune to find those cells that shouldn't be in our body. So the first thing that we had to show the US FDA when we presented them with our product was, particularly in cancer patients, that we could get blood from natural killer cells from cancer patients to grow equally well as if we were to draw blood from healthy subjects. It would be a shame to enroll a patient, particularly an end-stage cancer patient, into a trial only to be told some 2 to 3 weeks later that you could not get their natural killer cells to grow. So the FDA really asked us to show that we could do this consistently. And this is the part you can see on the left when we draw blood from healthy subjects, and on the right when we draw blood from heavily pretreated cancer patients, that we could get their NK cells to grow equally well. And this is something that a lot of companies really have not been able to do, and why they have gone straight to an allogenic product. The next thing is, even if you are able to get NK cells to grow, if you don't make the NK cell inherently stronger and you just put back in, let's say, 1 million weak NK cells, we would argue that that would not necessarily be biologically active for the patient. So you can see above is just when we draw blood from healthy subjects or heavily pretreated subjects, how we're able to get their cells to grow. But down below, the dotted line represents if I was to draw anyone's blood, isolate their resting natural killer cell, and plate it at various ratios against the leukemia cell line. If you go all the way to the right at a ratio of 0.5 to 1, meaning we took 5 of somebody's NK cells against 10 leukemia cells, you can see that even when we draw blood from healthy subjects, you don't get much killing. But after we've had a chance to activate and expand NK cells without any genetic modification, you can see that these highly enhanced activated NK cells at the same ratio kill a dramatically more number of cells than the patient's NK cells could beforehand. And we think this is very, very important to have very high natural killer cells that can, with high cytotoxicity, that can really carry out the natural intent of what natural killer cells were created to do. And this is just showing you from our current phase 1 cancer trial in the United States that when we drew blood from heavily pretreated cancer patients, you can see how much more powerful we made their natural killer cells compared to the historical donor average when we drew blood from healthy subjects and measured their resting natural killer cells in sort of the activity that I just showed you on the previous slide. But if we can do this for heavily pretreated cancer patients, we believe we can do this for anyone. The next thing is, unlike T cells, which rely on one receptor and as a result can cause a lot of collateral damage in the human body, our NK cells in all of us have 40 plus receptors, some that are activating, some that are inhibitory, but it's these receptors that are almost like a quality control mechanism. So as they police your body, they can, with all these sensitive receptors, determine whether or not that cell is normal and should be left alone, or whether or not the cell has some disease such as cancer or is virally infected, or it's a protein or some inflammatory cell that is causing damage. Our NK cells have the ability through all these receptors to seek them out and then destroy them. And again, this is just showing you that when we take blood from healthy subjects on day 0, that many of us have some receptor expression, but by the time we're done with our activation expansion process, not only have we resulted in billions of cells that have very, very high cytotoxicity, but now we've dramatically increased the activating receptor expression so that when these cells are put back into a patient, one, they don't cause any collateral damage because they know to leave normal tissue alone, but they're able to actively seek out those things that shouldn't be in our body and now have the killing potential behind it to then eliminate those cells or things that should not be in our body. Our process entails a peripheral blood draw where we can take as little as 100 milliliters of peripheral blood. We isolate the natural killer cells, and through our proprietary expansion activation process, we can produce large numbers of cells and doses, and we can freeze multiple doses that can be thawed for subsequent use. So I just want to at least point out that there are 2 different processes that people will talk about. Autologous is where we draw blood from the patient and give them back their own cells. Allogenic is where you use a universal donor, whether it be fetal-derived cord blood or pluripotent stem cell-derived NK cells, and those are given to a patient that's different than the original donor. What we really want to talk about in the context of using our NK cells for neurodegenerative diseases is the autologous product, and we'll explain why we think the autologous makes more sense in neurodegenerative diseases in just a few more slides. So one of the things we really want to talk about is neuroinflammation, which is sort of taken a back seat to all of the other processes or theories such as amyloid or alpha-synuclein protein hypotheses in Alzheimer's and Parkinson's. But really, the neuroinflammatory angle is something that we think is very, very important and is sort of underappreciated up to now. And if you see, neuroinflammation plays both a positive role in our body in terms of the ability to remove pathogens and things, but also there's very negative aspects of neuroinflammation, particularly when it's chronic, that can lead to neurodegenerative disease. It can lead to anxiety and depression. It also can lead to cognitive impairment. So we think that we can really help through the role of natural killer cells reduce the negative aspects of neuroinflammation. Now, I really first wanted to talk about the research that's been done in Alzheimer's and Parkinson's, and specifically how we feel our natural killer cells play a much more complete, comprehensive role in tackling both of these diseases, and how the diseases in terms of mechanism of action are very, very similar. So as we go back to the idea of neuroinflammation, there's been data to show that whether it's the amyloid tau proteins in Alzheimer's disease or the alpha-synuclein proteins in Parkinson's disease, that as these proteins accumulate in the brain, they elicit a whole full slew of neuroinflammation, and specifically how that neuroinflammation really starts to cause a lot of alterations in the crosstalks between glial cells and neurons. Of note is that two separate institutions have found that whether they be the alpha-synuclein proteins or the amyloid proteins, that when you take mouse models and you knock out the natural killer cells, either you deplete them or you work with a mouse that has had the natural killer cells knocked out, that causes an overwhelming acceleration of protein deposition. And both of these independent papers have concluded that natural killer cells can not only help to slow the deposition, but in some respects can also help to eliminate protein deposition in both of these conditions. The challenge is obviously immunosenescence. As people get older, the natural killer cells and in general, the whole immune system tends to get weaker. And this also allows for acceleration of protein deposition. What we also know about how these proteins then elicit a robust cascade is you can see several papers have now found in Parkinson's as well as Alzheimer's disease that they specifically elicit an autoreactive T cell autoimmune phenomena. So as these T cells are honing in towards these proteins, in fact, what they're doing is causing a lot of collateral damage to the surrounding neurons and glial pathways. And this is as much causing a lot of the changes and impairment in individuals as the protein is in and of itself. We would also argue that if you're just removing the proteins, as a lot of the current clinical trials have done, and still leaving the inflammation behind, that you may not see real robust improvement in the underlying cognitive impairment. And so we think in addition to removing the proteins, you also need to address this underlying inflammation. The other thing that's really interesting is to see the amount of autoimmune disease associated with dementia and Alzheimer's. Specifically, they found that if you had an underlying autoimmune disorder, which is again where your T cells are overreactive and causing damage to, let's say, the white matter in multiple sclerosis, or the joints in rheumatoid arthritis, or the skin in psoriasis, or the bowel inflammatory bowel disease, that they found you had a 33% increased risk of developing Parkinson's disease. And then also when they looked at patients with autoimmune disease, they found a large population were more likely to develop Alzheimer's disease. So again, this underlying imbalance in autoregulation of your existing T cells seems to predispose that inflammation, and could also be one of the variables that lead to a greater incidence of neurodegenerative diseases. This is work from Eric Vivier, where he looked at patients with active autoimmune diseases, and when they drew blood and looked specifically at natural killer cells, what was really interesting was that almost all of these autoimmune diseases were associated with either reduced numbers of natural killer cells or natural killer cells that had impaired activity, meaning they had weakened cytotoxicity or activating receptor expression. And why we believe that this is important, and how natural killer cells really support the overall avoidance of autoimmune situations, is that let me back up a second. So if you look at immune checkpoint inhibitors, which have become the sort of the new holy grail in oncology, what they do is they unleash the good and the bad of T cells to then attack the tumor, but they have associated collateral damage. One of the ways they do this is to cause an increase in the number of autoreactive CD8 T cells. And these T cells, while they are good at attacking the tumor, the ones that are in circulation can cause significant damage. So you can see that with the best-selling immune checkpoint inhibitor drugs, there's a very high incidence of severe bowel issues, skin issues, and endocrine issues. And that's because these autoreactive T cells are in fact damaging the healthy subjects, the healthy parts of the body that are not cancer. And that's one of the collateral damages that occur. What's interesting is we have been doing a trial in the United States using our cells in combination with these immune checkpoint inhibitors. We believe that it can further enhance the positive anti-tumor effects. As you can see here, we've treated somebody, 32-year-old, that's PD-L1 negative, who had been on Keytruda before. All those dark spots represent tumor, and after several doses, you can see how the tumor has melted away with a re-challenge. So this patient didn't respond to Keytruda before, but when we added Keytruda in our cells, you can see the patient responded. Down below is a patient who had not responded to Opdivo, which is another immune checkpoint inhibitor, but after we added our NK cells, we were able to see dramatic improvement. So on one hand, we saw improved anti-tumor activity, but what was really nice is we didn't see any side effects. So whereas you would see up to 20% grade 3 severe GI toxicity with Keytruda and up to 10% grade 3 severe toxicity with avelumab, when we combined our cells with either of those products, we saw 0 side effects. And this is because natural killer cells have been found to identify and eliminate these autoreactive T cells as part of a quality control mechanism. For all the intention that has been placed on regulatory T cells, it's actually that NK cells are really the ultimate regulatory T cell. The way they do this is through 2 receptors, NKG2D and DNAM1. You can see down below when we expand and activate and multiply our cells, not only do we increase the number of cells, the killing potential, and the activating receptor expression, but you can see here how we get almost 99% DNAM1 expression as well as NKG2D expression. Now, if you look over on the right, in a normal healthy situation, natural killer cells have the ability to find and identify and eliminate these autoreactive T cells through DNAM1 and NKG2D. You can see above the healthy situation, but down below, as we get older and the NK cells get either weaker in terms of their ability to kill or have less DNAM1 and NKG2D activating receptor expression, either they cannot recognize these autoreactive T cells well, or they don't have the killing potential to kill them once they do recognize them. So again, when we put our cells into our patients, they have very high DNAM1 and NKG2D, so they can identify not only tumor cells and other cells that shouldn't be there, but they can especially identify and eliminate these autoreactive T cells. And we believe this is the biggest reason why we have not seen any side effects when we combine our cells with the immune checkpoint inhibitors. And we think this is very important when we look at how we will treat neuro patients. The final thing I just want to give background on is that natural killer cells have also been shown to have the ability to identify and eliminate damaged neurons. So when you look at whether it be due to inflammation, whether it be due to protein deposition or infection, as you have damaged neurons there, they can greatly interfere with the ability for normal brain function by reducing the connectivity, the electrical impulses and such. And while we're not saying that the brain can necessarily regenerate new cells, we do think that as NK cells eliminate some of these damaged neurons, that it allows alternative signaling pathways to form. And so to just summarize so far, that we think our natural killer cells can not only help remove proteins, they can help to cool off the brain by removing these autoreactive T cells. And the final part is they can start to really eliminate these damaged neurons. Now, one of the questions people have is, how can you say that your NK cells can cross the blood-brain barrier? There are some natural killer cell products that are out there in clinical trials where they have to directly inject their NK cells into the brain tumor bed because they don't seem to cross the blood-brain barrier. Well, this is a study that showed the role of a chemokine receptor called CXCR3. And what that is when they looked at patients with really chronic brain issues, whether it be multiple sclerosis, Alzheimer's disease, Parkinson's disease, they found that those areas of the brain that were affected had very high levels of CXCR3. And more importantly, when they looked at the T cells in those patients, they also had very high CXCR3 expression. So the feeling is, particularly in Alzheimer's patients, that they found CXCL10 positive astrocytes, which were frequently associated with amyloid deposits. So you have amyloid deposits in those areas that have high CXCL10 expression. The T cells have very high CXCR3 expression that can cross the blood-brain barrier. And in fact, when they did autopsy data, where they looked at patients with chronic Alzheimer's disease and autopsies and stained the brains, they found that those specific areas, the neocortex, the hippocampus, the striatum, and the cerebellum, had very, very high expression of CXCR3. And that's, in fact, where all the proteins seem to accumulate and where you lose activity on a PET scan. And the idea is that you had very high protein deposition that led to very high CXCR3 expression. And then the T cells also had the CXCR3. So they crossed the blood-brain barrier, went specifically to those areas, and started to cause damage and inflammation due to the protein accumulation. And the same thing that you'll see in Parkinson's disease is this very high amount of CXCL10, CXCL11, and the CXCR3 that I just mentioned. Of note is when we activate and culture our cells, you can see from day 0 to day 17 how we get a very, very high CXCR3 expression. And when we've done studies looking at cell migration, our ability of our NK cells to go where the action is, you can see that when we look for CXCL9, CXCL10, CXCL11, that in fact our NK cells had high migration ability to go to those areas. So one of the ways that we would say that we feel our NK cells can cross the blood-brain barrier is specifically through this CXCR3 expression, that they're basically following the T cells to where the action is, and then going after the T cells based on the mechanisms that we discussed on the previous slides. So when we look at how our NK cells work, we think our NK cells first cross the blood-brain barrier versus CXCR3, and then through the presence of DNAM1 and NKG2D, they can identify and eliminate autoreactive T cells, while at the same time helping to secrete interferon gamma to activate macrophages and microglia to then help to eliminate the alpha-synuclein or amyloid proteins. The final thing that they can do is also through NKG2D identify and eliminate some damaged neurons. So it's a much more comprehensive way that NK cells function in the brain compared to just removing a protein. So we wanted to test this out on some patients. And while most of the clinical trials focus on mild cognitive impairment, we really wanted to take patients with really end-stage disease to see if we could make some improvement, and that there would be no doubt as to why the patients got better. So the first patient we came across was a 38-year-old when we met him, but at the time he was diagnosed at the age of 34 with advanced Alzheimer's. He was a Stanford a grad who was working in the Bay Area as a programmer. He kept getting lost on his way to work. Eventually was found to have a PSEN1 mutation at UCSF, which basically guaranteed before the age of 50, he would have full-blown Alzheimer's disease. When we met him, he was already in hospice requiring 24-hour care. You can see his PET scan that showed striking decreased activity, which was consistent with Alzheimer's disease. We actually reached out to Dr. Ming Guo, who's the head of Alzheimer's and Parkinson's at UCLA. Might she be able to see this patient before we treated him? She saw him and said this was probably one of the worst cases that she had seen. When we met him, he could not talk. he could not get out of a wheelchair and he could not feed himself. So after obtaining informed consent and discussing this with the family, who agreed to allow us to treat him, we proceeded to give him several doses of his own enhanced cells. Now, remember, when we met him, he could not walk, talk, or get out of a wheelchair.
All right, Dan, let's go. You have to unbuckle your seatbelt. All right, Daniel. Don't forget, you gotta what? What do you gotta do? There you go. Nice.
So not only could he now get out of the car, which I would suggest is more difficult than getting out of a wheelchair, he was then able to comprehend that he needed to close the door. Here's him eating.
Daniel, how's the food? Good. What are you eating? You like it? Yeah. Awesome. You want to try eating with yourself? Yeah. Okay. Do you remember how to use a chopstick? Yeah. Okay. Well, let's see.
This is him. The nurse took a picture of him in his nursing home while he was sleeping. And you can see his leg was trembling as if things were reconnecting and reawakening. So again, here's one example of a patient that was very, very end stage, was really not given any other options, and who has had a dramatic response.
Daniel, how's the food? All right, Daniel, let's go. Good. What are you eating?
Next is a patient of the head of capital markets at one of the leading banks, who I met at a conference several years ago. He reached out to me and said, you know, my mom has advanced Alzheimer's disease. Do you know anything or anyone that's doing anything innovative? We mentioned to the family that we had something that was not yet proven, but that we would consider making it available for her. We had started her treatment pre-COVID in a country that considers a non-genetically modified autologous treatment as a treatment, not a drug, and does not require FDA approval. But then COVID hit, there was a 2-year lapse, and we were unable to treat her again until last week, where the US FDA granted us approval to bring her treatment to the United States and resume treatment under compassionate use. What's really interesting is when we first met her, she didn't remember her kids' names, her husband's name, that she lived in New York, or any of those things. But here her husband, who has given me permission to share this video, talks about the experience.
Can you describe really what happened, her first experience with this?
The first six treatments?
Yeah.
We saw a significant benefit in her cognitive abilities. On a personal test, it went from a score of 8 to 32. And then we did 2 other tests after those visits. The score remained high for 6 months, but was declining and continued to decline when we stopped. We then did some testing before this treatment today, and we saw a score of 6 or 7, and we're looking forward to testing.
So here you have somebody describing his wife's experience before. And again, we just got approval from the US FDA to resume her treatments after a 2-year hiatus. And we're very anxious to see if we can get her improving again. And that will be the first question. But this is part of our larger goal to get a full USA approved IND and begin clinical trials in the United States in the next few months.
Can you just...
This is another patient that was treated in Japan, who's from Korea. Japan is another one of those countries that views this as a treatment, not a drug. And you can see she went from a scale of 12, which is severe dementia, to a score of 22, which is mild dementia. We have also treated somebody with advanced Parkinson's disease who has had marked improvement as well. Dr. Guo at UCLA saw this patient pre and post treatment and did also confirm that this patient had dramatic improvement. We have a current trial underway for advanced Alzheimer's disease. Unlike most of the trials are looking at mild cognitive impairment and trying to slow progression, we're trying to take really advanced Alzheimer's disease and show real disease modifying improvements. We have signed an agreement with the Parkinson's Foundation, and what they're really helping us to work on accelerating the path to get approval to do a Parkinson's trial in the United States. And really what I wanted to now talk about was the potential role of our natural killer cells in autism spectrum disorder, as well as the rationale of why we should consider it. First is that there are lots of publications looking at immune dysfunction in kids with autism. One specifically looked at natural killer cells in autism, and they found that kids with autism spectrum disorder had much weaker natural killer cells compared to cells from kids without autism spectrum disorder. On the right is also a study that looked at natural killer cell levels in autism clinics. It's a little bit misleading. So what this is saying is the number of kids, the average number of percentage of kids in an autism clinic that had weak NK cells was much, much higher than from a regular pediatric practice. So you already start to see some correlation with weak natural killer cells and autism in two separate publications. You also see another third publication that looked at the gene expression and function of peripheral blood natural killer cells in kids with autism. Again, found that it was weaker. And there's been a lot of great work from Judy Vanderwater and her staff that have looked at immune dysfunction in autism spectrum disorder, and they specifically found altered NK cell and T cell activity and populations in the autism spectrum population. What was also of note is that when they looked at adults with high-functioning autism spectrum disorder, and they isolated the natural killer cells and really looked at how well they could recharge and do their job, they found that they were very, very weak and got exhausted very, very quickly. We think this is important because even if some kids may appear to have normal natural killer cells in terms of the numbers, if you are to repeatedly challenge them, we think they'll be found to be very, very weak in terms of ability to continually kill and recharge. Another great series of research has been done by Paul Ashwood and his team that has also, again, looked at the role of immune dysfunction. They found dysfunctional natural killer cells, increased inflammatory cytokines, and the areas that at least we're focused on are the dysfunctional natural killer cells and the microglia activation that is all part of this chronic inflammatory process in kids. And that's the area that we think our natural killer cells could address because these kids also have weak underlying natural killer cells. Previously I'd mentioned that with Alzheimer's and Parkinson's, there was a correlation with increasing amounts of autoimmune disease. And in kids with ASD, the same thing. There was a study that looked at a total of 3,325 kids, and a third of them had infantile autism diagnosis, and they were found to have a much, much higher incidence of autoimmune disease, or moms with a maternal history of rheumatoid arthritis or celiac disease. This is just something that is consistent. There's also a study that looked at autoimmune diseases in kids as a whole, but they found that kids with ASD had a much higher incidence of autoimmune disease, and the most common autoimmune diseases that these kids had were either inflammatory bowel disease or psoriasis. And if you look from in the upper right-hand corner, patients with psoriasis are found to have very, very weak natural killer cells with reduced cytotoxicity and reduced cytokine production. So we do think that there are very big similarities in terms of the autoimmune component and weak natural killer cells that exist in ASD with some of the other neuroinflammatory diseases as well. This was a great paper that was done from Jun He and his team in Boston that again looked at neuroinflammatory conditions, particularly that happen in utero and the role of how this affects the inflammatory T cells. And what they concluded was that maternal IL-17A sometime in utero plays a big role in the inflammatory response in kids with ASD. What's really interesting about the IL-17 angle is that on the oncology side, IL-17 is known to be one of the big negative contributors towards natural killer cells in tumors, that this seems to suppress the anti-tumor effects of natural killer cells. So here you see IL-17 being reported in the oncology standpoint, but it does seem to weaken those natural killer cells. And as Dr. He and his group showed, that's a prevalent cytokine that's occurring and seems to contribute to the pathogenesis of ASD. So when we look at neuroinflammation during pregnancy, there are a lot of things that can cause inflammation within the mom, whether it be a high-fat diet, autoimmune disease, infection, pollution. One way or another, it causes some maternal immune activation, probably an increase in that IL-17 that we just mentioned, as well as TNF-alpha, IL-6, and this leads to some inflammatory process within in utero that can then further lead to full-blown neuroinflammation and the development of ASD. And we think that when the natural killer cells are weak and cannot help to regulate those T cells that are causing that inflammation, that's part of the process that is leading to this ASD. And so if you have to focus on the areas of neuroinflammation, that impaired immune regulation leads to increased inflammatory cytokine production, alters neurodevelopment, and increased potential for autoantibody production. And while NK cells cannot really affect that in utero, we do believe that as that inflammation starts to continue in the newborn, that those NK cells could very much help to cool off this inflammation. And also in patients with chronic neuroinflammation, that NK cells could help to play a role in this. Now, same type of thing. How do you know that the NK cells can go to the brain of children with autism? This was a study that looked at the upregulation of those chemokines that I had mentioned before. They found very high increased production of CXCR3 again, which again can be one of the ways that T cells are attracted to go to the brain and cause the inflammation. But if we were able to follow that with NK cells that have very high CXCR3 expression, they could very much traffic the blood-brain barrier and go after those T cells to help reduce that inflammation. In fact, this was a paper that came out in 2020 that said that one of the therapeutic opportunities that could be used for treating neuroinflammation would be to block CXCL10 and CXCR3. Well, if it's already there, we could then use our NK cells specifically through this pathway. And another study showing that this chronic inflammation can lead to increased seizure in epilepsy. That specifically, again, it's the CXCR3, and this is something that what they said was most of the conditions are associated with an increased incidence of seizure epilepsy, meaning those that had high inflammation were more susceptible. And then this was a recent study that just came out last week that showed that the neuroinflammation microenvironment, it basically reduces the anti-seizure threshold, and that this was one of the other areas that was probably increasing the risk of seizures. So when we think about our cells, we think, again, our cells can do several things. In those diseases that are initially protein-based, they can help to either slow the deposition or even improve the removal of those proteins. But at the same time, they tackle other issues such as the neuroinflammation and the damaged neurons that exist. Our NK cells can cross the blood-brain barrier. And what we've shown from all of our phase 1 cancer trials is there are no side effects associated with it. We've now dosed over 100 patients across 4 clinical trials without one grade 2 or grade 3 toxicity. We've had a few grade 1 patient-reported fatigue, which it's hard to say was really due to the cells or more due to their underlying advanced diseases. But we do think that the safety profile as well as the scientific rationale lends itself for us to try the use of this across numerous neurodegenerative diseases. And we're very excited to work with several leading organizations to bring this to the clinic for various indications in this coming year. Thank you for your time.